Hello, everybody. We'll go now over some of the multiple sequence alignment coloring methods for protein. As we said in the last lecture, there is some tools, some coloring methods for DNA, and we talked about them in the last lecture. And now we'll talk about the other ones that are for proteins. There is many coloring methods for protein, and you can just go to this multiple sequence alignment viewer application homepage, and you can read about them in much more details. You can also upload your protein alignment from that link, for example, in the multiple sequence alignment viewer, and also you can select any option and observe the change to the display. This is another way to learn about them. So by reading about them, you will find there is many ways for protein coloring. And by the way, these methods also available in different programs, like what we will talk about them later. Something like Jalview, for example. You will find many of these conservation and many of these ways for coloring. You will find them also in Jalview and other programs too. So, okay, we'll talk about the next first one. The first one is called conservation. This method, as we can see, highlight conserved and less conserved amino acid positions based on the relative entropy entropy threshold of the residue. All the alignment position with no gaps will be colored. Red indicates highly conserved, and the blue indicates the low conservation. As you can see here, there is two colors. This is the highly conserved, like this one's, these columns here at arrow number 1. This red colors is the most conserved, and this blue is the less conserved or lower conservation is here in this blue one at arrow number 2. Of course, there is also here, there is red here, more conserved. This column is most conserved, highly conserved, and this one is lower conserved. So you can see the difference very easily, and you can spot the conserved amino acids very easily and differentiate them from the lower conserved amino acid. Another method called small amino acid colors, and this is the default coloring method for protein alignment in the MSA viewer. And you can read about them here from that link here, and you can just open from here from this window. You can open that window, and you can see this menu is open by just pressing here in coloring. At arrow number 1, there is this menu called coloring. And you will find that the check mark here, this check and the small amino acid color, which means that this is the default coloring method in the MSA viewer. This small amino acid color renders of the 20 standard amino acid residue, of course, as well as ASX and GLX in a certain color. Along with one additional color for anything else, indicating nucleotides, for example, solvent, and non-amino acid ligand. Some colors are shared by 2 or more amino acids with similar properties. You can see them here, all of them, in this figure, residues of amino acids and colors, and which one have its color, and you can see the difference between each amino acid and the coloring, as you can see here. As we said in the briefly, there is some of them are sharing the same color, as you can see here. So here another one is called Blossom. Blossom methods are, as well as substitution. Blossom methods use well-known substitution matrices to display the degree of match of residue relative to each alignment position or column. And in this one, you can see here that we have two colors, of course. When an anchor row is set, the coloring in the column shows the match score to the residue on the anchor sequence. You have two, as you said, there is the blue. Blue represents a better match, and the second one is green, and green represents the worst match. So you have two colors which represent the extremities. The one is the blue color for better match and green for the worst match. When an anchor row is not set, the color reflects the average match of overall the other residues in the column. So you have two options. You can set an anchor row and then as we said, the blue represent better match to the anchor row, and green represent the worst match to the anchor row. This is number one. Number two, if the anchor is not set, the color reflect the average match over all the other residue in the column. So this way of coloring routines called Blossom. Okay, and I think Blossom also you can find it in many. By the way, you can find it in many other programs, as you can see in Jalview, for example. This one is very common to be used. It's called show differences. Show differences highlight differences compared to the anchor sequence also. In the alignment, you must set. You must in this one you must set an anchor row to use this option. And of course, they said notice please discard any coloring when unsetting the anchor row when still while still in the show difference mode. This is very important note that can be used, but I just want to emphasize and that this method can highlight, as you can see here, the color is red. You can see here the differences between compared to the anchor sequence. So you have like, well, for example, wild type, and you would like to compare sequences that may have mutation to your wild type. So, and this method is efficient and easy to, you can just put where the difference is because it just have the red color to see, to show you the differences. This one also good, it's called frequency-based difference. As we said, this is for protein. Frequency-based difference compare the residue at the position to the position column consensus. And darker shades here you have darker shades of red indicates a further difference from the residue in the other rows in the alignment at that position. So if you see here, you will see the different levels of shading and of the red color. So this is not just a red color like the last one, but also this one have like a degree of red color. The one is which is darker shades, this indicates a further difference from the one that have a little bit lighter red. So as you can see here, this one is a little bit light red, this one is a little bit darker, much more darker. So that's why this the difference is much more. So you can see and compare between the residue at the position at the same column, as you can see. Here another way of coloring, this is called hydropathy scale. This is different from the other ones because this coloration method reflects the side chain hydropathy with the one with very hydrophobic in red and the one that hydrophilic in blue. So this is different because this is only concerned about hydropathy scale. So this is hydropathy for the type of the amino acid that is hydrophobic in red, and the one is more very hydrophilic will be in blue, as you can see here. And you can, of course, read more about that one from that link if you want. Here is the membrane preferences. This is also special one. This coloration method reflect membrane buried preferences. Parameters as described also that's in that paper if you would like to go and read more about it. The color ranges from the red, which is have a low membrane preferences, and green, which is has a high preferences. So this is the difference between both of them. Here is structure prediction of membrane bound protein. As you can see, there is a shapely amino acid color. This way, shapely is another traditional coloring method. Shapely colors are described also here if you would like to read more about them. This is also a special coloring method for a special case, as you can see. Shapely uses different set of colors for amino acid, each one different, and also color differently by the six nucleotides. Also, so this one, Shapely, can be used for, there is a coloring scheme for protein and that method, and also this coloring scheme by that method for the DNA and nucleotides. Signal sequence, also this special one, this coloring reflect the signal sequence helical potential as described here. So this coloring method, as you said, it's only for the signal sequences, or much more used much more in the research for signaling peptide and signal sequences in your sequence. So this method also a special case for the people who are interested in this such kind of research for the signal sequence coloring, and to just try to indicate where these are, and you can read much more about them here in that link. Size, and this is also special one. This color from the red to blue, but here we reflect the size of the amino acid. So this will be based on the size of the amino acid. The red means the representing the smaller side chain, the smaller amino acid, I mean. So of course, because the smaller, it has a smaller side chain, so that's why it will be much smaller. And while blue is the larger side chain. So the people who are interested to differentiate between the structure of different proteins based on the size, if you would like to see based on the size, the best way to color your proteins based on this, because it's only this method reflect the size of the amino acid, size of the amino acid, and it has two colors, red and blue. Of course, you can, after your color, making your multiple sequence alignment and color them based on the what you want, what method you want to color them with, and based on your research and based on your interest and what you would like to show the people by coloring them, you can just download your alignment just by pressing here in the printer-friendly PDF or SVG here from the downloading button here, which exists here. You can download and press download. You can see, of course, before you can preview here if you would like to preview, and you can download by pressing just download here. So I hope it was easy. You can practice much more, and you can grab some practice using these links. Thank you very much for listening, and we'll go to the next lecture. Thank you. Bye.